All right, guys, let's go ahead and get started. This is chapter 15. Chapter 15 is going to cover innate immunity. There's two kinds of immunity in your body, innate and acquired. That's essentially it. We do have a first line of defense. It isn't really the immune system, but we'll mention that guy too. This is the basic breakdown of your entire defensive system relating to this whole entire thing. And you can see here we have a first line of defense on the far left, then the second line of defense, which is what we're mostly going to be talking about today, and then the third line of defense for the acquired, and what that is and what all of this stuff does, we're going to be getting into in greater detail in this chapter as well as the next chapter. So remember, this chapter is going to be covering innate immunity. It's kind of like the guys at the front line in a war. We know that we need to send somebody there to fight off the bad guys. So we send in the Marines, right? We send in the ground troops. But because the planes, the bombers, they're not here yet, right? Even if we call them in, they're not going to be there right away. The tanks, we call them in, they're not going to be there right away. So those guys are our acquired immunity. B for bomber and T for tank. I don't know. But basically the innate is the guy that's holding down the fort, holding down the line until those other guys show up. And man, when those other guys show up, the show is over. So our that's what we're getting into today. That's our innate immunity, the guys that are holding down till the tanks and the bombers show up. So the whole point of our immune system is basically to surveil everything in your body all the time, constantly looking for antigens and trying to destroy anything that we see as foreign. We have to then be able to recognize the difference between self and non-self. How do we recognize between self and non-self? Well, I'm glad you guys asked. That's going to be the surface markers on the cells. We've already mentioned a little bit about our surface markers, that viruses have spikes that can use those surface markers as a means of the lock that their spike key fits into, right? Those surface markers have normal functions for the cell. They might be a channel that allows for passages of ions. They might be some sort of docking mechanism for that cell to interact with another cell, or special situation sort of molecules that are on the surface. Whatever it is, whatever those molecules might be on the surface of our cells, we have to train our body to see that as self. This is a problem because just because I see that molecule in my body as self doesn't mean that I would see that molecule in your body as self. Now, maybe, there's not a whole lot of that going on of my body needing to recognize your body until you donate a kidney to me, until you donate, any sort of organ to me, maybe even part of your liver. It's less specific then, luckily, but it's an issue. So all those markers that are on the surface of the cell can be seen as foreign if they're not exactly the same as yours. But we have so many of them. Our body does a great job for the most part of training our immune cells to see them as self instead of non-self. So we're not attacking our own cells all the time. Now, of course, I'm sure you can hear where this is going, that it doesn't always work perfectly. We've already introduced what antigens are. I'm going to move on. Anything that we could, react to with our immune system, antigens. So the antigens, those surface markers are antigens, right? Any marker that would be anywhere that we're reacting to is an antigen. You can also call antigens immunogens. Yeah, that's all the same stuff. Okay. Okay, so it's a problem because if we aren't recognizing, first of all, self as self and non-self as non-self, then you might start a reaction against self when you're not supposed to. When you activate that whole route, it really can't be undone. So what happens is you get development of autoimmune disorders. Okay? There might be all sorts of different ways that can get activated. It could be a genetically related thing. It could be a combination of genetics and environment. It could be some environmental thing, and a lot of times it can even be from having some other disease. And then you react to that disease, and now you've got MS, for example. So that's how autoimmune disorders would work. We're going to talk about those in more detail in chapter 17. So we know how they're going to recognize self as self, those recognizable markers that we've trained our immune system to see self as we have grown up. How do we recognize the bad guys as bad guys though, right? And this is the pathogen associated molecular pattern. Absolutely going to be on the test. I'm going to ask you what PAMP stands for because we're going to call them PAMPs a lot. So you will need to know what each letter stands for. Pathogen associated molecular patterns, because lucky you, on the test there will be choices that all start with PAMP. So I'm not going to make it that easy. But yeah, so PAMPs. They're just like, okay, we have bacteria. We know our cell walls on our bacteria are made of peptidoglycan, right? It's just repeating sugar units linked together by peptide groups, right? So it's nothing crazy, but that is a repeating molecular pattern that we can see automatically as being associated with a pathogen. Peptidoglycan is a PAMP. That's all that we're talking about here. So just molecules that when your body sees them, it just instantly says, that's a bad guy, not supposed to be here. Start to fight now. It isn't very specific. So many things can have these markers on them, but if we see them, we know bad guy. All right, so we're going to get into what's important in the different aspects, or I guess, I don't want to say structures, but I guess aspects is a better way to look at it. But the things that participate in how our immune system works. For example, one, oh geez, this pen, I forget that it does this. One, mononuclear phagocyte system, or MPS. We'll come back to it. We don't need to worry about it right now. Number two, we're going to be talking a lot about the extracellular fluid, or ECF. This is literally just the fluid in the spaces around tissues and stuff. That space that is being filled with the fluid is called the interstitium. Number three, the bloodstream itself. This is how a lot of our lymphatic stuff is going to be transporting. Yeah, you might be sitting there being like, I thought that was like the lymph nodes and all that stuff. Yeah, it kind of is, but it's going to be complicated, of course. So we also have the lymphatic system, which is separate from the bloodstream. Yes, it's compartmentalized. They have their own vessels that are not part of the circulatory system. They have their own specialized accessory organs that are not part of the circulatory system. We'll get into the difference between these in a moment, all right? So these are the kind of main concepts of how our immune system is going to function, the structural basis of them. All right. The whole point of the lymphatic system, provide a means for the movement of our extracellular fluid, okay? It is sort of a drain off, and I like this metaphor, a drain off system for the inflammatory response. Let's say you stub your toe, right? Okay, guys, guess what actually happened? This is a real story. Okay, what actually happened this weekend on Sunday, we went kayaking, my husband and I like to kayak. I have 2 kayaks and whatever. We're putting the kayak back up, and he puts his side, he's on one side. We have these like straps to hang him up on in the garage. And he puts his side of the kayak with the straps turned around. So it won't sit right in the straps. And I'm like, well, you know, can you just turn that around? He's like, oh yeah. And he just pulls the strap off, and it was being held up by the strap, and it lands on my foot, right? Obviously, I mean, I'm wearing some heels, so it's not that big of a deal, but at the time it felt like a pretty big deal, right? So you know how it is when you like drop something heavy on your toes and you're not even really sure if they're broken or not, and all of this is pain that it's in. Your, toe might swell up. It's probably going to turn red. There might be some bruising, all of this stuff, the inflammatory response, right? The whole idea, though, is when you're having that inflammatory response and you're swelling up, that's because of that fluid, the extracellular fluid coming into that area, bringing in all of these signal molecules and ways for the cells to talk to each other. And the cells themselves that are going to be fighting all this fight, they aren't just always there. They have to be called in there. And when they do that, they come with all this fluid. And then when they're done, the fluid has to leave somehow. And as they're fighting, they might need to go out and interact with other cells. That drain off system is the lymphatic system. So when they leave, they go through the lymphatic system, not the bloodstream. They got there through the bloodstream. They leave through the lymphatic system. Okay. So that's how the gist of that is going to be working. Yes, and we will talk more about it in detail. I'm so glad that you guys are excited about it too. So the lymph, plasma-like fluid, it's just liquid that's going to be carrying everything related to our immune responses, as well as any debris and stuff like that, is going to be moving out from our extremities back into the circulatory system from an inflammatory response. So it will go into the lymph that way. So anything associated with that moves with it. So the white blood cells that were in the area, there could be fats, cellular debris from any sort of damaged cells in the area, nutrients from the area or that were going to the area. Infectious agents that had access to tissue spaces will now get moved through the lymphatic fluid because that extracellular fluid is pushing it into that drainage system. Okay, that's what the lymph is for. That's what the fluid is for. The lymphatic vessels themselves, their walls are very, very thin and are easily permeated by that extracellular fluid. That's going to be doing all that work in your immune responses. Okay, so it has to be able to cross easily. So they have thin, thin, thin cells there. So that's the vessels of the lymph system. So here's the biggest thing to remember about the lymph in general. Number one, lymph moves in one direction only, from the extremities to the heart. It is a return mechanism. Like we said, it's a drain off. So that drain off system is going back, just like your veins, whenever they're done doing their stuff, you have the deoxygenized red cells in your veins going back to the heart, right? That's what all this is doing too. But how does it get there? Your heart is not pumping your lymph. I've already told you it's not part of the circulatory system at this part. They've got their own vessels, completely different own vessels and their own organs at that point. So lymph is only going to be transported through the contraction of skeletal muscles. And if that sounds weird and like you didn't realize that, let me ask you something. Anybody in here ever have any surgery or serious injury to an extremity, like a leg or something? Okay, right. And then did they want you to walk on it pretty much as soon as you could? If you ever had surgery, they're like, okay, you're strong enough to walk on it. We need to start working on this. It's not just to build your muscles. It's to get your lymph flowing. And if you've had any kind of surgery at all, so I've had even my freaking tonsils out, they did this. I woke up from getting my freaking tonsils out and I had those things on my leg that like squeeze your legs alternately. This was at Baptist, right? So I don't know what they do at all the hospitals, but at Baptist they do. They put these squeezy things on your legs. That whole point is not to help your blood flow. It's to help your lymph flow because you can't get up and walk to move your lymphatic system around. And I'm telling you already, guys, your entire immune system relies on the lymph moving. And if it isn't moving, you won't have as great of a response to help heal yourself and everything like that. That's why they will tell you, get up and move. Like you had surgery, you feel like, okay, you're ready to go to the bathroom and walk. Okay, you're ready to walk then. We're walking, let's go. We're doing it. And you guys will push your patients to do the same thing because you will know now the importance of moving your skeletal muscles that will move the lymph. Now, we're not asking you to run marathons, right? Just move a little bit. Even if you're just sick, get up and move a little bit, maybe once an hour or something, make a run around the house, walk around the house once, and then go back and lay down. But getting that lymph moving is what's going to get your white blood cells moving too. And your white blood cells are the ones that are going to be doing the fighting. So lymph is only transported through the contraction of skeletal muscles, okay? And it only moves from the extremities to the heart. All that stuff that was going to the area was, again, coming through the bloodstream. They were moving there through the bloodstream, but now that they're coming back, they get their own special little highway to come back. That's because we need to filter them. We need to see what was in there. We need to check out what was going on at the inflammation site. Is there bad guys in there? Probably. So we want to check it out. This is just showing a diagram of where your lymph is. We're going to go through the organs of the lymphatic system. The red blood, red bone marrow, and the thymus are our primary lymphatic organs. So the red bone marrow, for those of you who don't know, that's where all of your blood cells are coming from. All blood cells. When I say all, I'm talking red blood cells and white blood cells. All right, they're all coming from that. Hematopoiesis, if you've heard of that, that's taking those stem cells in the red bone marrow and turning them into some kind of specific cell. Whatever it is, red blood cell maybe, white blood cell maybe, depending on your need, whatever. And then we have this structure called a thymus. Your thymus starts shrinking pretty much since the day you were born. It is the area where your T cells, which are part of your acquired immune system, are going to be maturing. So thymus stands for, T stands for thymus. Go figure. T cells, thymus cells. But basically they get their beginnings in the bone marrow, just like everybody else, and then move into the thymus for their maturation. A lot of that maturation is making sure you're not binding with self. Okay, so we can delete all of the guys that did want to bind with self. But anyways, so those are your biggies where we're going to have all the, origination from. Next phase, we have the lymph nodes, the spleen, and the alts. Okay, so we kind of know what a lymph node is a little bit. It's basically a hub in your lymphatic system where we're going to have concentration of your white cells hanging out, ready to interact with whatever is being filtered via the lymph. So it comes in through the lymph node, and the cells in there are going to come in basically like you have to have your passport. And if you don't have your passport at this checkpoint, that we're going to have a problem. So it's that sort of thing. That's your lymph nodes. And we have so many of them all over your body so that we can have checkpoints from anytime you get any inflammation from anywhere in your body. Then we have the spleen, and then salt, malt, and galt. The alts, we'll get into these. We're going to go through each of them. All right, the red bone marrow in your long bones. I kind of already told you that's where all our precursor cells are going, and all of your B cell maturation occurs in the bone marrow. It doesn't move out of there. Whereas the T cells, so those are our two lymphocytes for the acquired immunity. We'll mention those in a moment, but the T cells are going to mature in the thymus. The B cells just stay and mature in the bone marrow, and then they'll move to the lymph nodes. The lymph nodes, these are like it says bean-shaped organs. A lot of times we'll be feeling our neck and be thinking, oh yeah, lymph nodes, just like my tonsils, they're about the size of like a ping pong ball or something and whatever. If you're sick, yeah, but a normal lymph node from any other area, like where you're not actually sick, like let's say, yeah, okay, you have some sort of localized infection and they're swollen up. But if you go to a part of your body that there is no infection and you take a lymph node from there, your lymph node is about half the size of a pea. They're not very big. And you might be like, okay, so you think because you've seen pictures. I was a transplant immunologist at Baptist. I did all of the immunology work for any organs that were coming through at Baptist. And anytime that we had organs coming through from Baptist, the organs came with lymph nodes from the deceased donors. And then their lymph nodes, I would take their lymph nodes and I would remove all of their white cells out of those lymph nodes and wash them out and put them with serum and antibodies from the people who are going to receive it. So if their antibodies react with the cells that I got from the donors, they can't have that organ, right? And so it's like a 3-hour process when that happens. We also keep serum on file. If you are somebody who's waiting for a kidney, if you're on the list, we keep your serum on file every month and run it through our machines to see what antibodies in general you have so that we can match you up with the best match when it comes to be time, right? The time comes, I get called in at freaking 3 a.m. in the morning by myself, and I have to get these freaking lymph nodes from the dead guy. They give me like 2 from the whole body. I don't know how that works out, but it does. They give me like 2. And then I get the sample from the patient who usually is waiting in like that pre-surgery area, right? The prep area, waiting there. And the surgeon is waiting for me. And then I have this test that I have to run with these freaking 2 lymph nodes and my patient sample that you probably didn't give me enough of. And I have to run this test that takes 3 hours. And if that 3-hour test doesn't work, I have to do it all over again if I have the resource to do it. And sometimes you just don't, right? So no organ for that person. That's what that means. But I've never had that. I never had that happen when I worked there. I was terrified of it all the time, but it never happened. I always got my result and always was able to call the surgeon every single time because we had been doing our job ahead of time well and say that we're good to go with this organ. You're good to go. So we're not just waiting for nothing. I've never had it come back with a reaction because we do all this stuff ahead of time. But lymph nodes are tiny, y'all. They're tiny. I'm just saying. You can tell a lot about a person's background whenever they're tiny and they're black. If they're black like that, like they're usually like a yellow color, like a lot of the tissue inside of the body is, like fat sort of. But whenever they turn this blackish color, that means that they were smokers, like if they smoked like cigarettes. So I could tell if people were smokers from just their lymph nodes. So go figure. But not that I'm judging, I really am not, I promise. But yeah, so we know we have our aggregate. So those tiny little guys, those little lymph nodes are like the size of this little like eraser thing here. Hanging out in your armpit, hanging out in your neck. hanging out in your groin. These are the same lymph nodes that I'm talking about that get big that you can touch when you're sick and whatever. They are so tiny and they get so big because they're filling with all this fluid and all these white blood cells to kick everything into gear, to start multiplying our cells. Because the problem is we have somebody out at the extremities where you, I don't know, drop the kayak on your toe or something, or you have out, fighting, a disease in your lungs or something like this. Somewhere we need to get our B cells and T cells out there. But first, we have to call those troops in and start making sure they're all there and multiplying them and making sure we've got all the specialists and all these things in order and everybody's multiplied and proliferating and da-da-da-da. That takes a long time. So everything that you're experiencing when you are ill, typically, not always, but typically everything that you're experiencing when you're ill, if you're going to recover on your own, was your innate immune system, was working as hard as it could for you until your bombers and your tanks showed up. So we were waiting on them that whole time. So what, like for the most part, when you have a disease like that, when you're going to be sick and then getting over it on your own, like a week, right? So we're waiting about that long for these guys to show up. That's about how long it takes. But man, when they show up, I'll tell you this about when they show up. When your B cells and your T cells show up finally, these guys are fighting a good fight, don't get me wrong. When the B cells and the T cells show up, they do their job so well, they create memory cells afterwards, right? That's the whole point of a vaccine. It's not these guys, right? It's the B cells and the T cells. We're trying to create antibodies against something so that we can remember it. So when we see it again later, it works so well that if I brought somebody in here who actively had the measles and walked them up and down these aisles, you wouldn't so much as even cough within the next month, because your immune system has it so in check at that point. That's why we need the B's and the T's there as soon as we can. They can kick that disease out fast, but it takes a while to call them in. So anyways, lymph nodes is where your B cells are going to be hanging out for the most part. Your T cells will also hang out there in a slightly different area because your T cells, it turns out, have to activate your B cells. It just is a big old mess that we'll talk about in the next chapter. But everything is supporting that. Everything is supporting those. Innate immunity is just there to keep you together while we're waiting for the other part, okay? So that's why we have the swelling of your lymph nodes. Whenever those stuff gets activated, we're just multiplying those cells. The spleen, we know that the spleen, this is not on the lymph track, right? The spleen is on the blood track. We know it removes out our red blood cells. When your red blood cells are old, whenever they aren't functioning well or they have died, the spleen removes them. It also happens while it's doing that to be filtering out what's in the blood and has the phagocytes in its own tissue to remove any pathogens, anything that it sees as pathogens. Those phagocytes that are living out in the tissue and doing their job, those are called macrophages. We'll talk about more. Okay, these are the alts, the salts, the malts, and the galts. Okay, salt is skin-associated lymphoid tissue. Mucosa is mucosa-associated lymphoid tissue. And then we have gut-associated lymphoid tissue. And then we have tonsils and breasts whenever someone is lactating. There's a whole stuff that gets activated with that. But all of this stuff is lymphoid tissue where we'll have high concentrations of white cells, but they are not lymph nodes, right? So this has a similar function to the lymph nodes, but it's just tissue. It's not true nodes. This is why we have a lot of this stuff in the gut, specifically because of our microbes that live in our gut. So we have to know the difference between which ones are friendly and which ones are bad, right? All right. Okay. Anybody in here ever been a phlebotomist? Okay, so I just heads up, I was a phlebotomist at OBI for 3 years, so I used giant needles to draw blood for like hours at a time, sometimes up to 3 hours when I did platelets. And then I moved to DLO, where I was a phlebotomist for Southwest. And at Southwest, they're like lab workers, they're lab techs, also are phlebotomists. Like they do both at the same time. It's the weirdest DLO ever, but that's how I loved it. I loved being a phlebotomist. I liked talking to the patients or to donors when I was at OBI or whatever it was. I loved it. But I'm going to tell you something about the tubes. Whenever I get tubes in the lab, and you only send me this tiny, tiny little bit, and you're like, well, it's all I could get. I'm going to say, well, that's all I can't run the test from all you could give me though. So that's the problem. So if the lab is telling you they don't have enough, they're not lying to you. They're not trying to be mean. They're not trying to have you be like, they're not trying to be snooty or something like that. They will complain to you about this. That's why I'm saying it. Heads up. They absolutely will complain to you about the amount of blood that you send them sometimes. But they need a certain amount to run some of their tests. That's just how it works. Sometimes I have to send several milliliters of blood in for tests, like not just little drops amounts. People think that's all it is. It's not. Sometimes I need whole milliliters. I need multiple tubes for one test. But sometimes you're going to draw yourself tubes that have different colors of tops. The tiger top tubes, which are like the black and red spotted ones, if we call them tiger, I don't know, it doesn't look like a tiger to me, or the red top tubes. These guys have nothing in them. The tiger top had a little wax in there, but that's just a separator. That's all it is. So these guys, you're gonna send me that tube. You want to order your test stat. For some reason, you want a CMP run, and you ordered it with one of the red tops. And you ordered it stat. That's gonna be a problem, but okay, we can do it. So it has to sit there and clot first. So I can't tell you how many times we would get a stat tube in a red top tube that wasn't clotted, it was still warm from the patient. And then we'd get a call 5 minutes later on why they didn't get their results yet. And we'll be like, I don't know what to tell you, man. You sent me this tube. It wasn't me. I didn't ask for it. So yes, it has to actually clot to the point that when you tip that tube over, it is solid, right? It's crazy how it works. So you collect just blood by itself, nothing else mixed in it, clotted whole blood. You can then centrifuge that, and the clot part will settle at the bottom, and the liquid will separate out. And that liquid is called serum. Okay? It's not been treated with anything. There's no chemicals in it. However, you send me a green top tube or a blue top tube or something, or gold top tube. These all have things added into them to prevent clotting, right? They have anticoagulants in them. You draw blood with those. And if I spin that in a centrifuge, no matter how long it's been sitting out, if I spin that in a centrifuge, and then take it out and tip it over, it'll just mix back up again. So that is going to be completely non-clotted, completely unclotted. When you spin it in the centrifuge, you get those heavy red cells at the bottom and the liquids at the top. That liquid is called plasma. So plasma that has had some sort of anticoagulant is serum that has an anticoagulant introduced to the blood first. Okay, that's plasma. And the buffy coat there that it's pointing at in the picture, doesn't not a very good representation, but if you spin it correctly, you can get this white layer on top of the red cells, and that's the white blood cells. And there's just not very many in there compared to the whole volume, but they're what we're talking about today, some of the most important cells. Your red cells don't even have a nucleus. These cells fight. So they're way cooler than red cells, I'm just saying. Okay. Moving on, the white blood cells. All white blood cells are also called leukocytes. Okay, do not confuse leukocyte with lymphocyte. Leukocytes are our white blood cells, all of them. We differentiate them by staining them and looking at them in a microscope. They either have granules or they don't. These granules are just little like spots that are in the cell. They actually represent stuff that they can release to kill bad guys. But just because the agranulocytes, ones that don't have the granules, just because we can't see them doesn't mean that they don't do the same thing. We just can't see them in the stain. Okay, that's really how they differentiate these cells from one another. All right, there's some of these that are in this next picture that I have. We're going to have to know the difference between the granulocytes and agranulocytes. Some of them I don't need you to know, and some of them I don't need you to know right now. So I'm going to make it easier. Okay, first, on the very far right, we have our granulocytes. Don't pay attention to the man behind the curtain over here, okay? Our granulocytes, they stain as if they have granules in them. All right, up here, the ones that I'm gonna mention the most, especially for this chapter, the granulocytes that you can remember easily, the phils, okay? Neutrophils, basophils, eosinophils. If it ends in phil, it's definitely a granulocyte. Okay, all of them are granulocytes. Yes, I have mast cells down here. Okay, they are related more to allergies, and I'm not really concerned too much about their role in anything else besides allergies. So we will meet them and talk about them again in chapter 17 when we get into allergies. Okay, because I don't know if you know this, but that is your immune system doing things wrong, right? So anyways, That's your granulocyte. That's it, the fills. And I already broke this down for you with our lymphocytes, which are agranulocytes. Our agranulocytes, you can be divided into your lymphocytes and like your monocytes and the cells that come from monocytes. A monocyte is a cell that has a big old nucleus, no granules, that is able to do phagocytosis, and it's just floating around in the bloodstream. Once it moves into a tissue, though, it will differentiate in response to signals there. It will differentiate into macrophages. If it moves to the skin and hangs out there, they'll be dendritic cells. They're the same thing, though, really. Okay. Their functions are so similar, and there's not really any telling differences. I'm going to talk quite a lot about monocytes and macrophages, but especially macrophages. These are like the guys that are in charge of all the troops. Okay. But anyways, so our agranulocytes, we have the monocytes and their derivatives. The agranulocytes, we have the T cells and the B cells, the letter guys. Okay. The tanks and the bombers. Right. So we already know that those we're going to get to in the next chapter. Right. So agranulocytes, lymphocytes, monocytes, granulocytes, fills. Let's go into the fills. I don't care about the stuff at the bottom, by the way. All right, mononuclear phagocyte system. MPS. I told you we would come back to it. Yes. So the mononuclear phagocyte system, MPS, is just a whole bunch of fibers. They say it's reticular, but really it's just a transport system. So your cells can migrate between cells, between tissues, between organs. And so your macrophages and stuff, they don't have flagella. They're not swimming in it, all right? So they have to move along some sort of surface, if you will. So these fibers are something they can attach to and move along, okay? That's the mononuclear phagocyte system. It's just a way for them to move around in your tissues and between tissues. All right, our monocytes, right. So these are precursors to macrophages and dendritic cells. They're the ones that are hanging out in the blood, and when it moves into the tissue, they become one of those two. They can become specialized and hang out in tissues that we know that we can find them in, such as in the liver. The liver, those macrophages are called Kupffer cells. In the lungs, we have alveolar macrophages. I believe we've mentioned these before. These are the cells that tuberculosis actually likes to live inside of, these phagocytes. So that's the alveolar macrophages. Skin, we have the Langerhans cells, which are our dendritic cells in the skin. And then the brain, we have special cells that are very different from all these other ones. The microglia, they essentially work as macrophages with way toned down immune responses, because if you have a massive inflammatory response in your central nervous system, cause damage to your central nervous system cannot recover from that. It doesn't heal. So you're screwed. So we don't do it. So we have our own little kind of setup in the brain. All right, cytokines. Cytokines are just messenger molecules. They're going to tell the cells in the area what to do and where we need them and how to do it and all of this stuff, right? So there's 4 categories of these signal molecules. The pro-inflammatory, trying to cause the inflammation to start. The anti-inflammatory, trying to tell everybody to calm it down. The vasodilators and constrictors, controlling the blood flow and therefore the ability of things to move to and from the site. And then the growth factor ones. These are just going to help those B and T cells move faster, do their stuff faster, and encourage them to grow and come and hang out and do their job. All right, so this is just some examples of some of those cytokines, those signal molecules. You may have heard of some of them like interleukin or tumor necrosis factor. I'm sure you've heard of these vasodilators and constrictors that are involved in the inflammatory response, serotonin and histamine, right? Anyways, I don't need you to memorize these. I'm just bringing them up. So you've heard the terms and they're not alien terms anymore. We're going to start in by now breaking down the barriers themselves. First, starting with the physical and chemical barriers. Skin. The skin, the stratum corneum, exists of skin cells, dead, thickened layer at the top, and then, the mushy, nice, gooey layer underneath. But the thing that's making your skin waterproof and kind of impervious to things on the surface is keratin. And you might be arguing to yourself saying, my skin's not waterproof. Like waterproof gets on it. What are you talking about? Does that water that's on your hand or your arm or whatever get into the, muscle underneath it? No, because your skin is waterproof. So that's what it's talking about there, okay? Keratin is what is going to allow that to happen. the waterproofing kind of protein. Cool, cool, cool. Okay. Also in your skin, if I were to do this on my arm, and I don't have super dry skin today, if I were to do this enough, I'm sure you could see some skin cells flaking off. We've always got skin cells flaking off of our bodies. When those skin cells flake off, so do any microbes associated with them. So that's another way we can look at that. This stuff is literally me telling you about your body and how it just is there, and how come it works to protect you from microbes, even though it's not consciously doing that, and that's not its job. But hey, I'm going to tell you how it works anyways. The follicles, the skin glands, all of this stuff, either being shed or being flushing action, mucus membranes. We can have liquidy mucus or thicker mucus, catching the guys or flushing them out, whatever it is. The respiratory tract, we have a lot of mucus in that area, especially in the upper respiratory tract. We have nasal hairs to catch things, and the ciliated epithelium of your lungs are incredibly important. They're little hair-like appendages on the cells of your lungs that help move mucus out constantly. And what that does is you have mucus building up down in your lungs, and there's any bad guys that you're breathing in that's making its way down there. It moves the bad guys out, and then you cough, and then you swallow or you whatever, and then they're out. So that's the whole point of those. There's this disease that maybe you guys have heard of called whooping cough or pertussis. And whooping cough is also called 100-day cough. It is the type of cough that, okay, so babies really only get it now because people are vaccinated. But before you can get vaccinated, you are a baby, and babies cannot be vaccinated yet. So what happens is older people who are not up on their vaccinations might be carrying pertussis, go visit their little infant grandchild or whatever it is, give the baby pertussis. This happens in the United States. It's not unheard of. And then the baby gets whooping cough. This is the kind of cough. Like, have you ever had the kind of cough where it's like you're just coughing and coughing and coughing? You're coughing so much you just can't even breathe in. Like you're afraid like you might not even breathe again ever. That cough constantly, for 100 days. Well, not exactly, but you know, it's called the 100-day cough for a reason. As a infant coughing, imagine it. Imagine looking at a tiny little infant coughing like that can't even breathe in. That's whooping cough. So that kind of disease strips your lungs and your trachea and all of this stuff where the cilia are, strips it completely off. You don't have cilia really anymore after you've had pertussis. Like there's no way. And then after that, guess what? You're susceptible to pretty much any respiratory infection ever because you can't move it out of your lungs now. So that becomes an even more serious problem, as if it wasn't bad enough. Now your little infant who had recovered miraculously from this pertussis is going to have severe, severe, severe RSV or, you know, pneumonia or something. So it's all bad. So we need those cilia to move everything out. We need coughing to help with that as well. All right. The genitourinary tract, y'all know that you could pee. They tell you after sex that you're supposed to pee to help flush things out. That's this, okay? Vaginal secretions, we're also going to be mentioning here in a little bit about how, I'm sure you guys have heard, semen has antibacterial properties in it. Like there's like peptides in it or something that's antibacterial, whatever. Then we have our resident microbiota anywhere in your body, even especially in your gut. That's all going to be creating an unfavorable environment for your pathogens, meaning that they're going to take up the stadium seats. They're going to take up the nutrients. They have their own preferred pH that might not be preferable to the bad guys. All of this, the bad guys can't handle. That's called microbial antagonism. If you have ever had a tear or saliva, there is an enzyme in it called lysozyme. So this is not, you know, you've had all the ACEs, and I tell you about how that's enzymes. Well, this is a zyme. And if that zyme doesn't also say enzyme to you, I mean, it should. So lysozyme, it's an enzyme. It breaks down peptidoglycan, or a fancy word for that, hydrolyzes peptidoglycan. Remember, cell wall material of bacteria. So it can just break that stuff down. And our body is like full of it all over the place. So our tears, our saliva, and in your phagocytes, whenever we engulf the bad guys, we merge them with this toxic little ball of toxins. And one of the toxins in there is lysozyme. Heads up, that little ball of toxins, that little vesicle of toxins is called a lysosome. So, yeah. All right. Other chemical defenses, we talked about lysozyme. stomach acid. I mean, again, these things aren't made for this, right? They're just for other things, but happens to work in our favor. So you get the idea. Digestive juices and all this. All right, phagocytosis. I've been talking about it. This is where we're going to do it. This is where we really are. This is where it lives, all right? Phagocytosis, when we're going to be eating up our bad guys, we come up to it and we literally just take it in and make it part of our cell, put a little vesicle around it and hold it inside of us. The cells that mainly can do this, neutrophils, monocytes, and macrophages. Eosinophils can do it too, but I don't need you to know that. These are the big three, okay? Neutrophils are the most numerous white blood cell in the entirety of your blood, okay? The most numerous white blood cell by far. So they're kind of all over in your bloodstream compared to everything else anyways. So they're going to react very early on in the inflammatory with a high response. There's going to be a whole lot of them coming in. And they are, as it says, the primary component of pus because they act early. They have a lot of them there. They're the most in the bloodstream. So typically they're going to be the main component of pus, in addition to potentially any bacteria and debris in the area and fluid, of course, that moved into the area. So then we have eosinophils. Now, typically eosinophils are important for fighting off parasites. I don't know if any of you have allergies and have been found to have like elevated eosinophils. That was a problem. I have that problem like a lot, quite a lot actually. And it turns out that because we are so privileged here in the United States to not have many parasitic infections, our body doesn't really know what to do with the eosinophils. So the eosinophils, essentially, you get allergies because that's what they do now, I guess. I don't know. And we're going to talk about the role of antibody in allergies in chapter 17. Monocytes and macrophages. So I said it was neutrophils and monocytes and macrophages. Those are the biggies, right? Monocytes in the bloodstream. Once it moves into the tissue, macrophage. Monocytes in the bloodstream, macrophage in the tissues. Can't say it enough. Anyways, they'll get activated and move into these areas and eat things up in whatever tissue that they're called into to respond to the inflammation in that area, be part of the inflammation of the area. And what they will do is they can eat up whatever they eat up, the debris, the bad guy, whatever it is, break it down with those toxic lysosome vesicles. And what they have broken down, they can take and carry it around and take it over to the B and the T cells who are kind of just waiting, playing cards, waiting to get called up. And they'll bring this evidence and be like, y'all recognize this? That's what the macrophages and the monocytes will do. So basically go up to each of the cells there and be like, anybody here recognize this? And when one cell comes up and says, I do, now business is on with the B and the T cells. That is what a macrophage is good for. That's how the macrophages talk between the site of the infection where all this innate crap is going on and really get the big daddy guys activated and get that going because that's where it all happens, man. All right. Well, I know. Here we go. Yeah. All right. Here's the steps of phagocytosis. There's a picture here with more words on it, but basically we have chemotaxis. You guys remember what that is? We have some sort of chemical signal that has drawn something into an area. You could just be chilling and like smell the cookies down the hallway. And now you got to go eat the cookies, right? So I think I've used that analogy before. I guess you guys know I like cookies. But yeah, basically that's kind of what it's like. They get some sort of signal that tells them time to go this way. And as they keep getting it, they follow it and they follow that breadcrumb trail or cookie crumb trail and get to wherever they need to go. So we're here, we're at the site, we're at wherever this damage was done. The kayak was dropped on you. And we have, now we will start ingesting whatever is going wrong in that area. So any damaged cells, any debris in the area, any invaders, stuff like that, ingestion. Right, so we have them in our phagosome. Zome is just referring to a vesicle, okay? And then that phagosome will merge with a lysosome, again, vesicle, but the lysosome has all those toxins we've talked about. Not just lysozyme that breaks down the peptidoglycan, but also it has toxins like those toxic oxygen products that we've talked about in the past that some organisms can't have. It might even have acids or free radicals. All sorts of stuff can be inside of that lysosome. So when it merges with that bad guy vesicle, the phagosome, and becomes a phagolysosome, those chemicals mix with the bad guy and start killing it. So that's the destruction step. That takes about 30 minutes on contact. And then we will excrete all what's left over. Now, if we're macrophage or monocyte, we will also be breaking that down, taking some of the pieces and going around and being like, anybody know what this is? Anybody want to come out here and fight this crap? And that's basically what's going to be happening there. Okay, so that's how phagocytosis works and what its purpose is. I know we've been talking about eating stuff up this whole time. There's more stuff that happens after that. Basically eating it up and mixing it with toxins. Okay. All right, so PAMPs, back to these again. Let's say we're a macrophage that follows the cookie smell to the site. How do we know what we're supposed to be eating and breaking up and everything like that? How do we know if we should be trying to kill it? How should we send out signals that say that we need help? What drives all of that? The PAMPs do. So you come to this site where you smell the cookies and you come in there and you see that there's all these PAMPs around. So peptidoglycan, you can react to that. LPS, that is lipopolysaccharide. LPS, if you guys don't remember, that is also called endotoxin. And it's only found on gram negatives, okay, on their outer membrane. So we know if we see LPS hanging out in our environment, that's a gram negative, not supposed to be over in this area, right? Peptidoglycan, that's bacteria, it's not supposed to be here. Double-stranded RNA, we don't have that in our bodies naturally. So actual double-stranded RNA, we don't have that, right? We might have our weirdo tRNA or something, but that's as exotic as it gets. So if we have double-stranded RNA, that's truly double-stranded, 2 whole strands, that's not normal. That's viruses. It has to be. So that all of those can trigger our cells to be like bad, 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 bad. Time to set off the alarms. And then they'll secrete their own chemokines. It's calling even more guys to the area. Anyways, there's receptors that we don't need to know about. I forgot that that slide was in there. Okay, so we have our phagocytosis going on. We did our chemotaxis to the area, and we've formed our phagolysosome. If we ate up our bad guy, we ingested our bad guy. Based on the presence of the PAMPs, right? Made our phagolysosome here. That lysosome, toxin containing. The phagosome, the thing you ate and put the bad guy in, merging them together, okay? When that happens, we're breaking down the bad guy. It takes about 30 minutes to break down bacteria. We have things like the oxygen molecules, which we talked about before, toxic oxygen that the anaerobes can't handle. We have lactic acid, pretty normal, but the acid can skew the pH and make some things unhappy. Lysozyme, remember that breaks down peptidoglycan. And then nitric oxide. And there can be other enzymes and other molecules involved as well that can be deadly. All hanging out in that lysosome, just waiting to mix up, okay? So that, we've done that. We ate up our guy, we broke him down, and of course we're gonna get rid of the waste and maybe go talk to some B and T cells afterwards. So let's talk about inflammation itself. There's 4 words that you need to be familiar with. These are Latin, FYI, and so we'll start with rubor, or I'll start over here actually. Rubor, that's how I remember. If you don't speak Spanish, because I know a lot of the Spanish speaking folks here will recognize a lot of these words. I don't speak Spanish, so bear with me here. It's going to be kind of embarrassing. Rubor, I feel like rubor is easy to remember because it starts with R, redness. Okay, tumor. We know tumors are going to be like swollen up masses of flesh, just in general, tumors are. So tumor is a word that's talking about swelling. Okay, I'm going to move down to calor because I feel like this one is the most obvious one to me if you don't speak any Spanish. But to me, calor, you know what a calorie is. If you ever learned about calories, then you probably learned in chemistry, you should have learned about it, that it's like when you take like a milligram of sugar or something and you burn it, the energy that it releases is like a kilocalorie or something. We call those calories in the United States, but it's heat based. It's heat. The energy is released as heat. We call them a calorie because the Latin word calor means heat. So that calories, right? All right, and then the other one, we have dolor. This is pain. I don't have like a nice pun for you other than if you know the word. Okay, that's the only one. So cool. So those are the 4 terms, the Latin terms associated with the signs of inflammation. There's one more sign of inflammation other than redness, swelling, pain, and heat. And that's loss of function. Now, let me tell you, whenever I had that kayak dropped on my foot, it was like I couldn't walk on it. I was trying to be brave, trying to walk around, just, yep. Okay, we need to get the kayaks up. Can we just get the kayaks up? I'm going to put them up. That sort of stuff. And you're walking around trying to be all like tough and whatever. But you can't really step or put weight on your foot or whatever. That's the loss of function I'm talking about. It doesn't have to be massive, permanent loss of function. It could just be temporary because of the pain, because of the swelling, whatever. When I'm talking about the pain, I'm talking about the swelling, all this. This is inflammation. This is your immune system. This is what I'm talking about. Okay. Even just dropping a kayak on you, this is it. So that's inflammation. You can have chronic inflammation hanging out because of autoimmune disease or hanging out because of long COVID or whatever it is. But it can cause damage to your blood vessels, lungs, skin, and joints because these cells are being just, chronically activated to go fight this fight and, start spitting out all these molecules that are damaging. And it does damage your tissues over time. All right. Why do we have it then? What is the point of inflammation? So we're saying that it causes inflammation. And yeah, okay, so we have to get cells to the area. And you're telling me those are the good thing. But then also, what is inflammation and why do we have it? Okay. The whole point of inflammation, to mobilize and attract immune components to the site of the injury. So we get the heat and everything to start activating everything. We are expanding, vasodilating to move things into the area. So, bringing blood flow into the area. That's going to be that red color. We're trying to move all of our cells to come check out everything, start taking care of the damage and start fixing whatever just happened. All that is better done the more blood that gets to the area. Okay. We want to set in motion mechanisms to repair tissue damage and localize and clear away harmful substances. That's like, yeah, we're calling the cells in, we're calling the fluid in, but we need to be able to make sure they're doing their job, and that warmth of what is going on in your bloodstream and in that area and everything, and the fluid that's in there helps all that do its job there. Then we will destroy the microbes and block their further invasion. We do the best we can with this by eating them up and breaking them down. So let's go through our steps of inflammation. We have our injury. You stepped on a nail. It also brought in with it some bacteria. Probably did. This is a realistic scenario. Yes. So we've got our bacteria hanging out. The cells that are already there, ready to do something about this, they're just hanging. This happens to be mast cells. I know I said I wasn't going to talk about, but it just, in this case, happens to be. Okay. Whatever. These cells send out signals saying bad, bad, bad. This is bad. You see the bad? It's bad. The PAMPs are here. Okay. And see this vasoconstriction. I know I said about vasodilation, but in the beginning, vasoconstriction, because we're trying to keep everything compartmentalized. So we don't want blood that could potentially have bacteria in it getting out of that area. So vasoconstriction to prevent too much of that happening, badness moving out, okay? Now that we have received all these signals and we're ready to go do our job because there's PAMPs out there and we need to kill these bad guys, we've been called to the site from those cytokines, those chemicals. Now we will have vasodilation. We will now have our neutrophils moving in. They're going to be the first ones there, and they're going to be the most ones there. We're going to have liquid plasma from the blood, extracellular fluid, all that moving in with the neutrophils, containing everything that the neutrophils need, as well as just the cells themselves. And that's that swelling. We start forming a clot as well. Next, we have the edema and the pus formation. So we've got that buildup of fluid from bringing all those neutrophils in and doing their job. And now we're also going to need fluid to come in because we're going to start needing to move things out pretty soon. We're also still waiting on the B and the T's, right? The bombers and the tanks. So we have development of pus in these areas. This is just an accumulation of neutrophils, of the liquid in the area, of the bacteria itself, as well as dead cells, all that's in the area. That's pus. I mean, it really is just a mess of stuff in the area. But it's primarily in neutrophils, actually. And then number 4, finally, the lymphocytes. Remember, that's the word for both our B's and T's. Our lymphocytes have finally shown up to this area. And you notice that's just the end. There's no more story to this. So the macrophages that had interacted in the area can help activate all of this still as well and help eat up any trash that's being made from all of this, or any dead cells or whatever it is. But the lymphocytes are going to come in and do their job too. And that's just the end of it. Like there's no more fight that needs to be fought there. It's done. And yeah, you have a scar because your body doesn't care if you look pretty or not. It just wants to close it up, make sure you're safe. All right. So we do have vascular events that I don't need you really to know by detail, but just be aware of the fact that we do have vasoconstriction and vasodilation going on as appropriate for, whatever stage of the response we're in, depending on wherever your injury is as well, or illness, but whatever, it happens, is my point. Next, we have edema. This is going to include exudate, and that's going to mostly be stuff that you're leaking out of your wounds. Okay, so exudate, bloodborne components that escape into the extracellular space. So when I'm saying it weeps out, but that doesn't say, it just says in the space. Well, yeah, if you have an injury and you stepped on a nail or something like that, when you start having this oozy, clear stuff coming out of your wound, that is the exudate associated with your extracellular fluid moving into that area. If your bleeding has stopped, especially, then this is what you'll see next. It's that clear fluid, especially if you don't have like a whole lot of pus. But this can include pus. Okay. All right. So that's the exudate, and then edema is the swelling as a result of the accumulation of it. So if it is accumulating just under the skin and you don't have anywhere for it to go, it will swell up. That's edema. All right. I already said that we're going to send out signals. We're going to call in the neutrophils. The neutrophils are going to come and start killing things. And we're going to start this whole cascade of events that is going to really start getting rid of any bad guys or any damage or anything like this during our inflammatory response. How do the cells, though, moving in the bloodstream, get to the tissue site? So the bloodstream isn't quite like the lymph. It has a little bit thicker walls, but the endothelium of those capillaries in the blood are neutrophils and other white cells can squeeze through it. And that squeezing mechanism to change their shape and force their way into the tissue, wherever they're getting called to by chemotaxis, that's called diapedesis. It's just a movement squeezing through the cells of the red blood vessels. Yes. Anyways. White blood cells are going to respond to chemical signals and response to injuries or infection. We've kind of already been saying that like a million times. But what are the benefits of the swelling? What's the benefits of responding to all these signals? The fluid will dilute any toxic substances in the area. A lot of these microbes, these pathogens, produce toxins, unfortunately. So we can dilute that with the fluid. We can bring in the molecules needed to create fibrin clots to trap the microbes, as well as to prevent bleeding, to stop bleeding, and all of this stuff. Neutrophils, and we'll go around phagocytosing the bacteria and breaking them down, and that will lead to pus. Whatever their white waste is that they're making, the dead neutrophils, the dead bacteria, the bacteria themselves, the active neutrophils, all of this in the liquid makes pus. All right, that's the beginnings of it. Then our light reaction, finally, when we have our lymphocytes and our macrophages starting to get involved more in all of this. Yeah, so we're really coming in to clean up the area as macrophages, and our B and T cells finally get activated. Our B cells are going to make antibodies, and our T cells are going to kill the intruders directly or kill infected cells if we're talking about viruses. And we'll talk about those in detail, like I said, next chapter. All right. Part of the inflammatory reaction, especially if you are ill with something, it can be a fever. Okay? So this is an abnormally elevated body temperature, is a universal symptom typically of infection, though there are other things that can cause fever. Allergies, cancer, and other kinds of illnesses can trigger as well. I feel like I'm seeing something moving out of the corner of my eye. So I guess there were mice, though. I guess I might actually be seeing something. Whatever, ranges of your fever. I don't need you to memorize these ranges. This is something that maybe you guys would need to know if you do get into nursing school. I don't need you to know it, but to be aware, here are your ranges of your fever. Most doctors, when they meet you, if you are sick and you come and you're like, yeah, I've had a 99.2 fever, they might not visibly be rolling their eyes at you, but they are, okay? So you don't really need, if you are an adult and otherwise healthy person, if you have the temperature below 100, you don't need to be going for that, okay? If there's something else going on, go for that, but like coughing and coughing and coughing, fine, but this is not the reason, okay? If you get above 100 to 101, especially if it's lasting for a really, really long time, this is a concern. Moderate fevers and then high fevers. Of course, if you get above 102, I would hope you would be, concerned about your illness. There are chemicals in your body that can signal fever, but there are also molecules on bad guys that can signal fever as well. So if it comes from outside of your body, from the bad guys, let's say, the bacteria, the viruses, and whatever, or endotoxin, which again is just part of gram-negative bacteria, those are exogenous. So endotoxin is a potent pyrogen, in case I can't say that enough, because I'm pretty sure that's on the test. Endotoxin is a potent pyrogen. It causes fever. Pyro, think of fire, so that causes heat, fever. Endogenous, these are things that are inside of the body. You have some sort of chemical signal going on in your body that triggers fever. So this might be in response to an infection, but if your cells are making it, that's endogenous, okay? You can have both going on, of course, at the same time. All right, why though, right? I've already kind of mentioned a little bit about the benefit of having heat in an area. It helps activate everything, but here's why we have fever. It inhibits multiplication of organisms that are sensitive to temperature. Well, maybe they're not all. So does it help in any other way? Yes, it does. It impedes nutrition of bacteria that need iron because there are signal molecules in there that help with getting rid of the iron. This is associated with fevers. 3, increases metabolism and stimulates the immune reactions. Basically getting everybody active and helping everything along faster. So, for example, speeds up hematopoiesis, so making the new cells, phagocytosis, eating up the bad guys and breaking them down, and then adaptive immune reactions, so your B's and T's. Also increases the ability of specific lymphocytes to home in on the sites of infection. So it makes it easier for them to find where this is all going down. Okay? So fever does stuff. So here it is a difficult decision if you want to suppress it or not. You could say slight to moderate fever, you might let it run its course because it is doing something for your body. I am this person. I try to tough it out for as long as I can. It depends on how much pain you're in too, right? So that's another thing with it. But I try to tough it out where I can until I get to like 102. That's like my limit. If I get to 102, then I'll start taking medication. But yeah, so high and prolonged fevers are going to be risky and should be treated no matter what. And if you have anybody having any other symptoms caused by what seems to be from the fever, then you need to go to the ER. So you should also be considering anybody who has any sort of issues that would make them sensitive to the higher heat to take care of their fevers earlier. So it's saying if you have cardiovascular disease, head trauma, seizures, and respiratory ailments, you're going to be more sensitive to the fever as just a person. So consider that. My advice is simply that if you have a fever and you need to treat it, of course, with talking to your doctor, because I have to preface it with that, and I do mean it when I say talk to your doctor about it, but preferring when you can Tylenol over, Advil. Ibuprofen is, it can be really hard on your stomach, especially if you're taking it like consecutively. If you're just taking it once in a while, it's not so bad. But if you are taking it regularly, it's actually really severe on your stomach. I take a medication, for example. I take a medication where I'm absolutely not allowed to take NSAIDs because if I take it with my medication, I could be vomiting blood within hours. So nope to that. So if that can do that to me with that medication, otherwise I'm normal, right? That's enough. I came from a family that usually only took Advil, like took ibuprofen. So now I only have acetaminophen in my home because of that. But talk to your doctor, see what's best for you. Don't always lean on ibuprofen or other NSAIDs. See what works for your family. All right. Because remember, your inflammatory response is there for a reason. Okay. Antimicrobial products, interferon. We've already met interferon, but there's all different kinds. There's alpha, beta, and gamma. But this is the guy that I mentioned. This is when you have a virus coming, you have a burglar coming in your house. You go hide in your closet, and you are probably going to get killed. But you at least have time to call your buddy next door. So your buddy next door, here's your call that you're making. The buddy next door gets the message. So he goes and grabs his shotgun. That's what this yellow is about. He grabs his shotgun and he's ready to fight. So when the virus comes in, it has no chance. That's what interferon does. It interferes with that. It also can interfere not with just viral infections, but with cancer cells and things like that. And we can actually utilize that mechanism in treating certain diseases, especially when we're talking about cancer. Pretty cool. It also reduces apparently the amount of cholesterol in the body. That's literally the first time I actually read that. So there you go, something. All right, complements. This is the last little bit that we're going to go over. Complement. This is about 50 different blood proteins. This is literally just proteins that are always in your blood. They don't have to be activated or induced in any way. They're just always there, constitutive, right? They work together to destroy bacteria and viruses by this cascade reaction. Call it the complement cascade. Basically, one protein in there gets activated by its signal. We'll talk about the signals in a second. Gets activated by its signal, and maybe it is going to break in half or change its little shape. And then that leads to the next thing doing its thing, and the next thing doing its thing, and so on and so on and so forth. That's what a cascade reaction would be. That's simple. There's 3 ways you can activate that initial step, right? Activating complement. You can do it by antibodies. We call this the classical pathway. Classical pathway. Antibodies are around. If antibodies bind onto the thing, complement starts doing its thing, no matter what. Then we have the lectin pathway. Lectin is something that we have in our body that recognizes sugars on microbes. So basically, lectin pathway, recognizing sugars on microbes. And then the alternative pathway, this is going to be repeating molecules like LPS, things that like PAMPs. PAMPs could fall into here, okay? PAMPs in general, we see it as bad guy marker, then this is going to activate the alternative pathway. So LPS is endotoxin gram negatives, right? All right, so those are the 3 ways you can activate the complement pathways. No matter what, once it's activated, we go through this cycle of these proteins. And I don't need you to know the proteins, but I do need you to know what the end results are, okay? We can help with phagocytosis. There's a special word for this that we're going to mention later on, and when we talk about antibodies. But when we are helping out phagocytosis, we're basically putting a handle on the outside of the gooey like capsule. We know how many of those bacteria have the capsule. It makes it hard for phagocytosis. This puts a protein handle on the outside so that phagocytes can come along and eat up the bacteria easier. So that is going to help out with phagocytosis. Just FYI, the word that we're going to be introducing in the next chapter is called opsonization. That's what that is. Don't worry, we'll see it next chapter. Then we have inflammation. We've been talking all about this, but this can help aid in activating that, because sometimes this is the first thing that sees anything. If complement just hanging out in your bloodstream interacts with the lectins and doing the sugars on the bad guys, then this can start the inflammatory process, or at least aid in it. And then we have the membrane attack complexes. This is why complement is cool, okay? So we see this weird shape down here with the microbe, and we have this, I don't know, tunnel or whatever that is forming giant, but it's not supposed to be that big, but it's just for, so you can see it. But anyways, proteins and in the complement pathway come together and build this hole in the outsides of the microbes. And we have several holes in a microbe. So that's, we can get our chemicals in to fight them, but also their cell contents leak out. So just poking holes in these guys. That's the membrane attack complex. It's made of complement proteins. So there's 4 stages to all this that lead to it. Initiation, no duh, you have to start it somehow. So that's your classical and your alternative and your lectin pathways. Initiation, we got it started. We've got amplification and cascade. That's literally just all of those steps happening to lead to the phagocytosis, to the inflammation, to the membrane thing. Polymerization, bringing proteins at the very end of all this together to make that circle in the bad guy, and that's the membrane attack. Now we've done it, we've got the holes, and we can get our enzymes in, and they'll be leaking out their contents. Okay, so it's the 4 stages of the complement cascade. That's it. So that is chapter 15. So our monocytes important only for the innate immune response, or might they play any other important roles? We did talk about how monocytes and macrophages can break stuff up and then go around and be like, do you recognize this? To get everything else started for the B and T cells. That's a different kind of response. That's not your innate. That's going to be your acquired immunity. So, all right.